Welcome to DREAMLAB Knowledge, where tonight we embark on a journey into the most mysterious and magnificent object in the known cosmos, the human brain. Nestled within the protective vault of your skull, weighing merely 3 pounds, sits an organ so bewilderingly complex, so staggeringly sophisticated, that after thousands of years of human inquiry, we have barely begun to comprehend its depths. This mass of tissue, with the consistency of soft butter and the appearance of a wrinkled walnut, contains more connections than there are stars in the Milky Way galaxy. It orchestrates every thought you've ever had, every emotion you've ever felt, every movement you've ever made, and every memory you've ever stored. It is simultaneously the observer and the observed, the questioner and the questioned, the explorer and the territory being explored. As you settle into this moment of quiet reflection, consider the profound paradox. You are using your brain to contemplate your brain. The very organ that allows you to think about consciousness is itself the source of that consciousness. The neurons firing in your cerebral cortex right now, translating these words into meaning, are the same biological machinery attempting to understand their own existence. This recursive loop, this hall of mirrors where the mind examines itself, represents one of the deepest mysteries in all of science and philosophy. The brain is not merely an organ like the heart or the liver, performing a straightforward mechanical function. It is the seat of everything that makes you uniquely you, the biological foundation of human experience itself. Tonight, as we traverse the landscapes of neuroscience, neuroplasticity, consciousness, and cognition, prepare to confront questions that have puzzled philosophers and scientists for millennia. We will explore how 86 billion neurons communicate across 100 trillion synaptic connections. We will investigate how this biological machine generates the illusion of a unified self, the sensation of subjective experience, and the remarkable capacity for abstract thought that distinguishes our species. We will examine how the brain builds itself during development, how it adapts and changes throughout life, and how it can tragically unravel in disease. Along the way, we will confront the limits of our understanding and stand in awe before the magnitude of what remains unknown. The architecture of thought. Let us begin with the fundamental building blocks of this extraordinary organ. The human brain contains approximately 86 billion neurons, specialized cells designed for one primary purpose: communication. These neurons are not randomly scattered like grains of sand on a beach. They are organized into intricate networks, layered structures, and specialized regions, each contributing to the symphony of human consciousness. But raw numbers alone cannot capture the brain's true complexity. What matters most is not merely how many neurons we possess, but rather how they connect and communicate with one another. Each individual neuron is a marvel of biological engineering. Picture a neuron as a tree with a thick trunk representing the cell body, or soma, where the nucleus resides and metabolic activity takes place. From this central body extend branch-like structures called dendrites, the receivers of information, which stretch outward like the limbs of a winter oak, creating a dense forest of neural arbor. These dendrites are covered with tiny protrusions called dendritic spines, specialized structures where connections with other neurons are formed. A single neuron in your cerebral cortex might have 10,000 or more synapses, junction points where signals pass from one cell to another, each representing a potential pathway for information flow. But neurons are more than passive receivers. Each possesses a long, thin extension called an axon, which can stretch remarkable distances relative to the cell's size. Some axons in your body extend from the base of your spine all the way down to your toes, covering distances exceeding 1 meter. Along these axons, electrical signals race at speeds that can reach 400 feet per second, transmitting information across the vast neural landscapes of your nervous system. At the terminal end of each axon, specialized structures called synaptic boutons contain thousands of tiny vesicles filled with chemical messengers, neurotransmitters, ready to be released into the microscopic gaps between neurons. These gaps, called synaptic clefts, are astonishingly small, less than 50 nanometers wide, far thinner than a human hair. Yet across these infinitesimal spaces, the brain performs its most crucial operation, converting electrical signals into chemical messages and back again. When an electrical impulse or action potential races down an axon and reaches the synaptic terminal, it triggers the release of neurotransmitters into the cleft. These molecules drift across the gap and bind to receptor proteins on the receiving neuron's dendrite, either exciting it to fire its own action potential or inhibiting it from doing so. This electrochemical dance, repeated trillions of times each second across your entire brain, is the fundamental mechanism underlying every aspect of your mental life. The sheer scale of these connections staggers the imagination. In a single cubic millimeter of your cerebral cortex, a space smaller than a grain of sand, researchers have identified nearly 150 million synapses. Multiply this across the entire brain, and you arrive at estimates of 100 trillion to 150 trillion synaptic connections in the adult human brain. To put this in perspective, if you counted one synapse per second, it would take you more than 3 million years to count them all. During early childhood, at the peak of brain development, synapses form at a rate of approximately 10,000 every 15 minutes. This explosive growth creates a neural network of staggering complexity, one that makes the most sophisticated computer networks appear crude by comparison. But neurons do not work alone. They are supported by an equal number of glial cells, once thought to be merely the brain's scaffolding, but now recognized as active participants in neural processing. Astrocytes, star-shaped cells that wrap around blood vessels, regulate the chemical environment around neurons and help control blood flow in response to neural activity. Oligodendrocytes produce myelin, the fatty insulation that wraps around axons and allows electrical signals to travel at extraordinary speeds. Microglia, the brain's immune cells, patrol the neural landscape, pruning unnecessary connections and defending against pathogens. Together, these cells create an ecosystem of incredible sophistication, where each component plays a crucial role in maintaining the delicate balance necessary for thought and consciousness. The hungry brain. For all its computational power, the human brain operates on remarkably modest energy requirements, yet those requirements are absolutely critical. Despite representing only 2% of your total body weight, your brain consumes approximately 20% of all the glucose-derived energy your body produces. This disproportionate energy demand reflects the enormous metabolic cost of maintaining the electrochemical gradients necessary for neural signaling and the constant synthesis of neurotransmitters, proteins, and other essential molecules. Every moment of every day, your brain processes roughly 5.5 milligrams of glucose per 100 grams of brain tissue per minute. This glucose is almost entirely oxidized through a complex series of metabolic reactions, ultimately producing the cellular fuel molecule ATP, adenosine triphosphate. Each synapse in your brain metabolizes approximately 1 glucose molecule per millisecond, generating enough ATP to power the intricate molecular machinery of neural communication. The oxygen consumption required for this process is equally impressive. Your brain accounts for nearly 20% of your body's total oxygen intake, far exceeding what its small size would suggest. This metabolic hunger becomes even more pronounced during childhood. In a child between the ages of 4 and 8, the brain's glucose consumption reaches its lifetime peak, accounting for as much as 66% of the body's resting metabolic rate. This enormous energy investment reflects the intensive process of brain growth and synaptic remodeling that occurs during these critical developmental years. Young brains are not merely smaller versions of adult brains. They are metabolic powerhouses, furiously building and refining the neural circuits that will support a lifetime of learning and adaptation. The brain's energy demands are unevenly distributed across its geography. When you focus intently on a challenging task, engage in creative problem solving, or recall a vivid memory, specific brain regions light up with heightened metabolic activity. Blood flow increases to these active areas, delivering additional glucose and oxygen to fuel the increased neural firing. This localized increase in energy consumption forms the basis of functional brain imaging techniques like fMRI, which allow researchers to observe the living brain at work, mapping the neural correlates of thought, emotion, and perception. Yet even at rest, when you lie quietly with your eyes closed and your mind wandering, your brain maintains a remarkably high level of metabolic activity. This baseline energy consumption, once thought to represent mere neural housekeeping, is now understood to reflect the continuous operation of large-scale brain networks involved in self-referential thought, memory consolidation, and maintaining readiness to respond to the world. The brain, it seems, is never truly at rest. It is always humming with activity, even during sleep, maintaining the delicate electrochemical balance that makes consciousness possible. The living landscape of cortex. If we could peel back the protective layers of your skull and examine the surface of your brain, we would encounter the cerebral cortex, a thin sheet of neural tissue, merely 2 to 4 millimeters thick, yet containing approximately 14 to 16 billion neurons. This cortical layer has been dramatically folded and compressed to fit within the confines of the skull, creating the characteristic wrinkled appearance of the human brain. These folds, called gyri, are separated by deep grooves called sulci, and this extensive folding increases the brain's surface area enormously, allowing far more cortical neurons to pack into a limited volume. The cerebral cortex is organized into two hemispheres, left and right, connected by a thick bundle of nerve fibers called the corpus callosum. This massive commissure contains approximately 200 million axons that allow the two halves of your brain to communicate and coordinate their activities. Each hemisphere is further divided into 4 lobes, each associated with distinct functions, yet intimately interconnected with all the others. The frontal lobes, located behind your forehead, constitute the largest portion of the cerebral cortex and house some of humanity's most distinctive cognitive capabilities. Within the frontal cortex lies the prefrontal region, often called the brain's executive center, which orchestrates high-level decision-making, planning, personality expression, and the regulation of emotions and social behavior. This is the part of your brain that allows you to imagine future scenarios, weigh potential consequences, inhibit impulsive behaviors, and pursue long-term goals despite short-term temptations. Damage to the prefrontal cortex can dramatically alter personality and judgment, as famously illustrated by the case of Phineas Gage, a railroad worker who survived an iron rod being driven through his frontal lobe in 1848, only to emerge a changed man, impulsive, irreverent, and unable to stick to plans. Just posterior to the prefrontal cortex, separated by a prominent groove called the central sulcus, lies the primary motor cortex, which controls voluntary movement of your body's muscles. This region is organized somatotopically, meaning that adjacent areas of the motor cortex control adjacent parts of the body. However, this mapping is not proportional to actual body size. Instead, body parts requiring fine motor control, like the hands and face, occupy disproportionately large cortical territories. If you were to draw a human figure with body parts sized according to their cortical representation, you would create a bizarre distortion called a motor homunculus, with enormous hands, lips, and tongue, but tiny trunk and legs. Behind the central sulcus lies the parietal lobe, home to the primary somatosensory cortex, which processes tactile information from your skin, muscles, and joints. Like the motor cortex, the somatosensory cortex is organized somatotopically, creating a sensory homunculus that mirrors the motor homunculus. But the parietal lobe does far more than register touch. It integrates sensory information from multiple modalities to create a coherent representation of your body in space, enables you to perform complex mathematical calculations, and helps you navigate through three-dimensional environments. The temporal lobes, located roughly beneath your temples, serve as the brain's auditory processing centers, housing the primary auditory cortex, which analyzes the frequency, pitch, and location of sounds. Deep within the temporal lobe lies the hippocampus, a seahorse-shaped structure absolutely crucial for forming new memories. Without a functioning hippocampus, you would live in an eternal present, unable to convert short-term experiences into lasting memories. The temporal lobe also contains structures involved in recognizing faces, understanding language, and processing emotions, making it essential for social cognition and communication. Finally, at the back of your brain, occupying the smallest of the cortical lobes, lies the occipital lobe, dedicated almost entirely to processing visual information. The primary visual cortex, located around a deep fold called the calcarine sulcus, receives signals from your eyes and begins the complex process of analyzing edges, orientations, colors, and movements that ultimately allows you to see the world. From this initial processing area, visual information flows forward into higher-order visual areas that recognize objects, faces, and scenes, demonstrating how the brain builds complex perceptions from simple sensory inputs. But this division into lobes and regions, while useful for understanding broad functional organization, obscures a deeper truth. The brain operates as an integrated whole, with information constantly flowing between regions, creating dynamic patterns of activity that shift and evolve from moment to moment. Your experience of reading these words, for instance, involves not just your visual cortex, but also language areas in your temporal and frontal lobes, attention networks spanning multiple brain regions, and memory systems that connect new information with prior knowledge. The brain is not a collection of isolated modules, but rather a vast network of networks, each influencing all the others in a continuous dance of neural activity. The mystery of consciousness. Of all the phenomena the brain produces, none is more profound or more puzzling than consciousness itself. Right now, as you process these words, you are experiencing something. There is, as philosophers put it, something it is like to be you at this moment. You have subjective experiences, the sensation of light entering your eyes, the feeling of your body resting, perhaps the sound of ambient noise, the stream of thoughts flowing through your mind. These qualitative experiences, these raw feels of what it's like to perceive, think, and exist, are called qualia by philosophers and neuroscientists. The hard problem of consciousness, as philosopher David Chalmers termed it in 1995, asks, why should physical processes in the brain give rise to subjective experience at all? We can, in principle, explain how the brain processes information, how it integrates sensory data, how it controls behavior, and how different neural patterns correlate with different mental states. These are the easy problems of consciousness. Easy not because they're simple, but because we can at least imagine how to solve them through careful scientific investigation. But even if we had a complete neuron-by-neuron understanding of how your brain works, a fundamental question would remain. Why does this physical system generate the felt quality of experience? Why isn't all this information processing happening in the dark, without anyone home to experience it? Consider the redness of red, the specific qualitative character of what it's like to see a ripe tomato or a sunset. This subjective quality seems fundamentally different from any objective description of wavelengths of light, photoreceptor activations, or neural firing patterns. Even if you knew everything about how the visual system processes wavelengths of light around 650 nanometers, even if you understood every neurotransmitter release and every spike train in the visual cortex, you still wouldn't know what red looks like unless you had experienced it yourself. This is the essence of the hard problem. Consciousness seems to possess properties that cannot be reduced to or explained by physical processes alone. Various theories attempt to bridge this explanatory gap. Global workspace theory, proposed by cognitive psychologist Bernard Bars, suggests that consciousness arises when information becomes globally available to different cognitive systems throughout the brain. According to this view, information processed unconsciously in local brain regions can be broadcast to a global workspace, a network of widely distributed neurons that makes this information accessible to systems involved in memory, language, planning, and action. When information enters this global workspace, it becomes conscious. Prominent neuroscientist Stanislas Dehaene has developed neurobiologically detailed versions of this theory, identifying specific patterns of neural activity associated with conscious versus unconscious processing. Another influential approach is integrated information theory, developed by neuroscientist Giulio Tononi. This theory proposes that consciousness corresponds to integrated information that exists as a unified whole and cannot be reduced to independent parts. According to this framework, any system that integrates information in the right way possesses some degree of consciousness, with the quantity of consciousness measured by a value called phi. Systems with high phi, like the human brain, have rich, complex conscious experiences. Systems with low phi, like simple thermostats or even digital computers running current architectures, might have minimal or no consciousness. This theory makes the striking prediction that consciousness is an intrinsic property of certain physical systems determined by their cause-effect structure rather than by what they're made of or what they do. Yet another perspective comes from higher-order thought theories, which propose that a mental state becomes conscious when you become aware of that state, when you have a thought about your thought, so to speak. On this view, you might process visual information unconsciously, but the processing only becomes conscious when higher-order brain areas represent the fact that you're seeing something. These theories point to the prefrontal cortex and other association areas as critical for generating the higher-order representations that make first-order perceptions conscious. Despite these sophisticated theoretical frameworks, consciousness remains deeply mysterious. We still cannot explain why integrated information feels like anything, why global broadcasting creates subjective experience, or why higher-order thoughts generate qualia. Some philosophers and scientists argue that consciousness might be fundamentally irreducible, that it's a basic feature of reality, like mass or charge, that cannot be explained in terms of anything more fundamental. Others maintain that we simply haven't yet developed the right concepts or discovered the crucial neural mechanisms, and that consciousness will eventually be explained through ordinary neuroscience, just as life was explained through molecular biology. What we do know is that consciousness depends on the brain. Damage to specific brain regions can eliminate particular aspects of conscious experience. Injury to the visual cortex can create blindness, even with healthy eyes. Damage to the parietal cortex can make you unaware of one half of space or even of your own body parts. Disruption to the thalamus, a deep structure that serves as a relay station for sensory information, can eliminate consciousness altogether. The contents of consciousness, what you're aware of at any given moment, correlate with specific patterns of neural activity that can be measured and, to some degree, predicted. We can even use brain imaging to decode what someone is seeing or thinking about, at least in simplified laboratory conditions. Yet the relationship between brain and consciousness remains one of the deepest puzzles in science. We can observe that neural activity and conscious experience are correlated, but we still don't understand how or why the former gives rise to the latter. This explanatory gap persists even as our knowledge of the brain grows ever more detailed and comprehensive, suggesting that solving the mystery of consciousness may require not just more data, but fundamentally new ways of thinking about the relationship between physical processes and subjective experience. The brain that rewires itself. One of the most remarkable discoveries in neuroscience over the past several decades is that the adult brain is not a static, fixed organ, but rather a dynamic, changeable structure capable of profound reorganization throughout life. This property, called neuroplasticity, fundamentally challenges the old notion that the brain you're born with is essentially the brain you're stuck with. Instead, we now know that experience continuously shapes and reshapes the physical structure of your brain, forging new connections, strengthening existing ones, weakening those that are rarely used, and even, in certain limited regions, generating new neurons well into adulthood. The concept of neuroplasticity was revolutionary because for much of the 20th century, neuroscientists believed that once the brain matured in early adulthood, its structure became essentially fixed. Neural circuits, it was thought, could not be significantly modified by experience. But beginning in the 1960s and accelerating dramatically in recent decades, a wealth of evidence has demolished this static view, revealing instead a brain that remains malleable and responsive to experience throughout the lifespan. At the heart of neuroplasticity lies synaptic plasticity, the ability of synapses to strengthen or weaken over time in response to activity. When you learn something new, when you practice a skill, when you form a memory, what's happening at the cellular level is that specific synapses are being modified. A phenomenon called long-term potentiation, or LTP, describes how repeated, coordinated activation of a synapse can strengthen that connection, making it easier for signals to pass between the two. Neurons in the future. The neuroscientist Donald Hebb famously summarized this principle. Neurons that fire together, wire together. Conversely, synapses that are rarely used undergo long-term depression, becoming weaker and less influential. These changes are not merely functional, but structural. When a synapse strengthens, the physical size and number of receptors at that synapse can increase. The dendritic spine itself can grow larger and more stable, and new synaptic connections can form. When you learn to play a musical instrument, for example, the regions of your motor cortex controlling your fingers undergo expansion, with more cortical territory devoted to these frequently used movements. When you master a new language, structural changes occur in language processing areas. When London taxi drivers spend years memorizing the city's bewildering maze of streets, their hippocampi, the brain regions crucial for spatial navigation, actually grow larger. The brain's plasticity is most dramatic during childhood when neural circuits are being initially established and refined. During these critical periods, experience plays an especially powerful role in shaping brain development. The visual system, for instance, has a critical period early in life when visual experience is essential for normal development. If a child has a cataract or severely crossed eyes that aren't corrected during this window, the neural circuits processing vision may never develop normally, even if the optical problem is later fixed. This is because the brain's visual areas are competing for cortical territory. And if one pathway doesn't receive adequate stimulation during the critical period, other pathways take over that cortical space. But plasticity doesn't end with childhood. While it's true that the young brain is more malleable than the adult brain, adults retain considerable capacity for neural reorganization. People who lose their sight as adults often show remarkable cross-modal plasticity, with their visual cortex being recruited to process touch and sound. This allows some blind individuals to develop extraordinary abilities in other sensory domains. Stroke patients can sometimes recover lost functions through rehabilitation that leverages neuroplasticity, with undamaged brain regions gradually taking over functions previously performed by damaged areas. Even more striking, we now know that the adult brain continues to produce new neurons in certain regions, a process called neurogenesis. For decades, neuroscientists believed that neurogenesis stopped at birth, that you were born with all the neurons you would ever have, and that from that point forward it was a one-way street of neural loss. But in the 1990s, researchers definitively established that new neurons are born throughout adulthood in the hippocampus, the region crucial for forming new memories. These newborn neurons integrate into existing circuits and contribute to learning and memory. What triggers neuroplasticity? The most powerful driver is experience itself, particularly novel, challenging experiences that engage attention and motivation. Physical exercise has been shown to boost neuroplasticity, increasing the production of growth factors like brain-derived neurotrophic factor, or BDNF, which supports the survival and growth of neurons. Learning new skills, especially those requiring coordination and attention, promotes synaptic growth and reorganization. Even cognitive training and meditation have been shown to induce measurable structural changes in the brain. The dark side of neuroplasticity is that it can also work against you. Chronic stress can cause atrophy of dendrites and loss of synapses in the hippocampus and prefrontal cortex, regions critical for memory and executive function. Substance abuse can hijack the brain's reward circuits, strengthening pathways that drive addiction while weakening those involved in self-control. Chronic pain can lead to maladaptive plasticity, where pain circuits become hypersensitive, maintaining suffering even after the original injury has healed. Understanding neuroplasticity has profound implications for education, rehabilitation, and mental health. It suggests that our brains are not prisoners of our genes or our early experiences, but remain capable of change throughout life. The skills you practice, the knowledge you pursue, the habits you cultivate, all of these literally reshape the physical structure of your brain. This insight places agency in your hands. By choosing how to engage your mind, you influence what your brain becomes. The evolutionary triumph. The human brain did not spring into existence fully formed. It is the product of hundreds of millions of years of evolution, a magnificent and improbable outcome of natural selection operating on the brains of our distant ancestors. To understand the human brain, we must appreciate where it came from and why it evolved the particular characteristics that distinguish it from the brains of other species. The evolutionary story of the brain begins with the first nervous systems, which emerged in simple animals hundreds of millions of years ago. Even primitive organisms like flatworms possess clusters of neurons organized into rudimentary brains, allowing them to sense their environment and coordinate behavior. As life diversified and became more complex, so too did nervous systems, with different lineages evolving different brain architectures to meet different survival challenges. The vertebrate brain, which humans share with all other animals possessing backbones, has a basic organization that has been conserved for hundreds of millions of years. All vertebrate brains possess a hindbrain controlling basic vital functions, a midbrain involved in sensory processing and motor control, and a forebrain that in mammals has expanded dramatically to become the cerebral cortex. This three-part organization reflects our deep evolutionary heritage, connecting us to fish, amphibians, reptiles, and birds. What makes the human brain special is not a completely unique architecture, but rather specific elaborations and refinements of structures present in other mammals, particularly our primate relatives. The most obvious distinction is size. The human brain is roughly 3 times larger than what we would expect for a primate of our body size, a measure called encephalization. But size alone doesn't explain human intelligence. Elephants and whales have brains far larger than ours, yet they don't build civilizations, create art, or ponder the origins of the universe. More important than absolute brain size is the organization and connectivity of the cortex. Humans have approximately 86 billion neurons, which places us at the high end for mammals, but not dramatically above whales and elephants. What appears to set humans apart is the number of neurons packed into our cerebral cortex, about 16 billion cortical neurons, which is actually more than any other species studied so far, including species with larger total brain sizes. This high cortical neuron count, combined with efficient cortical organization and extensive long-range connections, may provide the computational substrate for human-specific cognitive abilities. The human prefrontal cortex, the region associated with planning, reasoning, and behavioral regulation, is disproportionately large compared to other primates. While representing about 1/3 of our total cortex, it constitutes a larger fraction than in any other species. This expansion of prefrontal cortex correlates with our enhanced abilities in abstract thinking, long-term planning, impulse control, and theory of mind. The capacity to understand that other individuals have their own thoughts, beliefs, and intentions. Language represents another distinctly human capacity that required specific neural specializations. While other animals communicate, human language is unique in its recursive structure, its capacity to express an unlimited range of meanings through combinations of finite elements, and its flexibility in discussing not just the immediate present, but the distant past, the imagined future, and abstract concepts. The neural basis of language involves specialized regions in the frontal and temporal cortex, particularly Broca's area and Wernicke's area, along with extensive networks connecting these regions to broader cognitive systems. But what evolutionary pressures led to these brain specializations? One influential hypothesis is the social brain theory, which proposes that human intelligence evolved primarily to navigate complex social environments rather than to solve ecological problems. Living in large social groups required keeping track of relationships, predicting others' behavior, forming coalitions, detecting cheaters, and coordinating cooperative activities. These social challenges, the theory suggests, created selection pressure for enhanced cognitive abilities, which then could be applied to other domains like tool use, hunting, and cultural learning. Another factor was likely the shift toward an omnivorous diet that included meat, which provided dense calories and nutrients to fuel a hungry brain. Cooking, which humans have practiced for perhaps hundreds of thousands of years, made nutrients even more available, potentially enabling further brain expansion. The development of tools, from simple stone implements to complex composite weapons, created a feedback loop where better tools required more intelligence, and more intelligence enabled better tools. Whatever the specific evolutionary pathways, the result is clear. Humans possess a brain that, while built on the same basic plan as other mammalian brains, has specific enhancements that enable uniquely human capabilities. Our brains allow us to create complex symbolic systems, like language and mathematics, to imagine scenarios that have never occurred, to teach and learn across generations through cultural transmission, and to contemplate our own existence and mortality. These capacities, emerging from billions of neurons, arranged in specific architectures shaped by millions of years of evolution, have enabled our species to transform the planet and extend our reach beyond it. Yet we should not fall into the trap of thinking humans represent the pinnacle of evolution, or that our form of intelligence is the only valuable kind. Evolution has no direction or goal. It simply favors whatever works in a given environment. Other species have evolved remarkable cognitive abilities suited to their own ecological niches. Corvids like crows and ravens display problem-solving abilities and tool use comparable to great apes. Octopuses, with brains organized completely differently from vertebrates, show sophisticated learning and memory. Elephants possess remarkable social cognition and appear to grieve their dead. Intelligence comes in many forms, and the human version is simply one variation on this theme, one that happens to have led us to build the tools and cultures that now surround us. When the brain breaks down, for all its resilience and adaptability, the brain remains vulnerable to a host of disorders that can disrupt its delicate functioning, sometimes catastrophically. Neurodegenerative diseases, in which neurons progressively malfunction and die, represent some of the most devastating conditions affecting humanity. These disorders strip away cognitive abilities, personality, memory, and motor control, confronting us with the fragility of the neural processes that make us who we are. Alzheimer's disease, the most common cause of dementia, affects millions of people worldwide, primarily those over age 65. It begins insidiously, with mild forgetfulness and confusion that might initially be dismissed as normal aging. But as the disease progresses, the symptoms intensify. Memory loss becomes severe, language abilities decline, familiar surroundings become confusing, and eventually, patients lose the ability to recognize loved ones, to care for themselves, to speak coherently. The final stages are marked by near-total cognitive collapse and physical debilitation. At the cellular level, Alzheimer's is characterized by two hallmark pathologies: extracellular plaques composed of beta-amyloid protein and intracellular tangles formed by hyperphosphorylated tau protein. These abnormal protein aggregates accumulate first in the hippocampus and entorhinal cortex, regions crucial for memory formation, then spread progressively to other brain areas. As they accumulate, synapses fail, neurons die, and brain tissue atrophies. The mechanisms driving this protein aggregation and neuronal death are still being unraveled, but they appear to involve complex interactions between genetic factors, age-related changes in cellular quality control, inflammation, and vascular dysfunction. Recent breakthroughs have produced medications that can reduce amyloid beta levels in the brain, marking the first treatments that target the underlying disease process rather than merely managing symptoms. While these drugs show modest benefits in slowing cognitive decline, they represent a significant milestone in Alzheimer's research and offer hope that more effective treatments may follow. Researchers are pursuing multiple therapeutic strategies, preventing protein aggregation, promoting clearance of abnormal proteins, reducing inflammation, protecting synapses, and enhancing brain resilience. Parkinson's disease, the second most common neurodegenerative disorder, primarily affects movement. People with Parkinson's develop a characteristic combination of tremor at rest, muscle rigidity, slowed movements, and postural instability. They may develop a shuffling gait, lose facial expressiveness, speak softly, and have trouble initiating movements. In later stages, many develop dementia as well, as the disease spreads beyond motor circuits to affect cognitive areas. The core pathology involves the death of dopamine-producing neurons in a deep brain structure called the substantia nigra. Dopamine is a neurotransmitter crucial for coordinating movement, and its loss in Parkinson's creates the characteristic motor symptoms. Like Alzheimer's, Parkinson's involves abnormal protein aggregation, In this case, clumps of a protein called alpha-synuclein that form structures called Lewy bodies within neurons. Current treatments focus on replacing lost dopamine or stimulating dopamine receptors, but these approaches don't stop the underlying neurodegeneration. Intriguingly, researchers have discovered substantial overlap between Alzheimer's and Parkinson's. Many Alzheimer's patients show some Parkinsonian features, while many Parkinson's patients develop dementia. At autopsy, significant numbers of patients with one disease also show pathological features of the other. Some scientists now propose that these conditions might represent different manifestations along a spectrum of neurodegenerative processes rather than entirely distinct diseases. This perspective suggests that insights into one condition might illuminate the others, and that therapies developed for one might prove useful for several. A third major neurodegenerative disease is Lewy body dementia, characterized by cognitive decline accompanied by visual hallucinations, fluctuating attention and alertness, and Parkinsonian motor features. Like Parkinson's, it involves alpha-synuclein aggregation and Lewy body formation. But unlike typical Parkinson's, the pathology is more widespread in cortical areas from the disease's onset. Amyotrophic lateral sclerosis, or ALS, targets motor neurons, causing progressive paralysis that is almost always fatal within several years of diagnosis. Huntington's disease, caused by a specific genetic mutation, leads to involuntary movements, cognitive decline, and psychiatric symptoms, typically beginning in midlife. What these diseases share is the gradual, relentless destruction of neurons and synapses, often associated with the accumulation of misfolded proteins that spread through neural circuits. They remind us that the brain's normal functioning depends on exquisitely precise molecular quality control, synaptic maintenance, and cellular housekeeping. When these systems fail, whether through aging, genetic vulnerability, environmental insults, or some combination, the consequences can be devastating. Beyond neurodegenerative diseases, the brain is vulnerable to stroke, trauma, tumors, infections, developmental disorders, psychiatric conditions, and countless other pathologies. A stroke can destroy the language centers in minutes, leaving someone able to think but unable to speak or understand words. A traumatic brain injury can alter personality and judgment permanently. Schizophrenia and autism spectrum disorders emerge from alterations in brain development and connectivity, profoundly affecting how people perceive and interact with the world. These disorders, tragic as they are for those affected, also serve as cruel natural experiments that illuminate how the brain normally functions. By studying what happens when specific brain regions are damaged or specific neural systems malfunction, neuroscientists gain insights impossible to obtain from studying healthy brains alone. Each patient, each case study, each pathological variant teaches us something about the brain's organization and the neural basis of human capabilities. Dreams of the sleeping brain. As you drift toward sleep tonight, your brain will not rest. Instead, it will embark on one of its most mysterious processes, the generation of dreams. Sleep is not simply the absence of wakefulness, but an active brain state with its own complex architecture. Throughout the night, your brain cycles through different stages, from light sleep to deep, slow-wave sleep to rapid eye movement sleep, where dreams are most vivid and frequent. During deep sleep, your cortex exhibits slow, synchronized oscillations, with millions of neurons firing in coordinated waves. This rhythmic activity is thought to play crucial roles in memory consolidation, moving information from temporary storage in the hippocampus to more permanent storage in the cortex. The sleeping brain replays experiences from the day, strengthening important memories while allowing less significant ones to fade. This is why sleep is so crucial for learning. The consolidation that occurs during sleep is often as important as the initial encoding during wakefulness. REM sleep, named for the rapid eye movements that characterize this state, is when the brain becomes almost as active as during waking, yet you remain deeply asleep. During REM, your brainstem actively paralyzes your muscles to prevent you from acting out your dreams. The content of dreams during REM sleep tends to be vivid, bizarre, and emotionally intense, often involving impossible scenarios, sudden scene changes, and illogical events that your dreaming mind accepts without question. The meaning and purpose of dreams remain subjects of intense debate. Are they random neural noise, or do they serve important functions in emotional regulation, memory processing, or creative problem-solving? The brain across time. Your brain today is not the brain you were born with, nor will it be the brain you have decades from now. The brain develops, matures, and ages, changing dramatically across the lifespan in ways both predictable and variable. A newborn's brain is remarkably immature, containing most of the neurons it will ever have, but with far fewer connections between them. The infant brain is primed for learning, with periods of explosive synapse formation that far exceed adult levels. A young child's brain contains far more synapses than an adult's, by some estimates 50% more. This synaptic exuberance provides raw material for learning, but it must be refined through experience. The brain selectively strengthens frequently used connections while pruning away those that are rarely activated, sculpting neural circuits through the experiences of childhood and adolescence. The prefrontal cortex, the brain's executive center, is one of the last regions to fully mature, with development continuing into the mid-20s. This protracted development helps explain why adolescents and young adults often show impulsive behavior, difficulty with long-term planning, and heightened emotional reactivity. Their prefrontal cortex is still establishing the connections needed to regulate behavior and emotion effectively. As we age beyond young adulthood, the brain begins a slow, gradual decline in some capacities. Processing speed slows, working memory becomes less efficient, and the ability to learn some types of new information diminishes. Brain volume decreases slightly, particularly in the frontal cortex and hippocampus. Some types of memory, particularly episodic memory for specific events, become more vulnerable to disruption. Yet aging is not a story of universal decline. Crystallized intelligence, accumulated knowledge, and expertise can continue to grow throughout life. Older adults often show enhanced emotional regulation and wisdom in navigating complex social situations. Many cognitive abilities remain stable or even improve with age, particularly if people remain intellectually engaged and physically active. The aging brain retains substantial plasticity, capable of forming new connections and adapting to new challenges, though perhaps not with the same facility as younger brains. The frontier of understanding. We stand at an extraordinary moment in the history of neuroscience. Never before have we possessed such powerful tools to observe and manipulate the living brain. Advanced imaging techniques allow us to watch neural activity in real time as people think, feel, and remember. Optogenetics enables researchers to control specific neurons with light, turning them on or off at will to determine their causal role in behavior. Brain-computer interfaces can decode neural signals to move robotic limbs or type words on a screen, offering hope to people with paralysis. Yet for all these advances, fundamental mysteries remain. We still do not fully understand how memories are encoded and retrieved, how the brain generates the unified experience of a moment, despite processing occurring in distributed circuits, or how consciousness emerges from neural activity. We cannot yet cure Alzheimer's or Parkinson's, cannot reliably treat depression or schizophrenia, cannot restore function after severe brain injury. The brain is sometimes called the most complex structure in the known universe, and this is not mere hyperbole. The 100 trillion synaptic connections in your brain far exceed the number of stars in our galaxy. The patterns of activity flowing through these connections create an inner universe of experience whose richness rivals the external world. Your brain is simultaneously a biological organ, an information processing system, and the substrate of consciousness itself, three aspects of one remarkable entity that we are only beginning to understand. The two minds within one brain. Before we conclude our journey, we must explore one of the brain's most fascinating organizational principles, the division of labor between the two cerebral hemispheres. Your brain, despite appearing as a single unified organ, is actually composed of two distinct halves, each with its own specializations, its own style of processing information, its own contributions to your mental life. These two hemispheres are physically separated by a deep fissure running front to back, dividing your brain into left and right halves. Yet they are intimately connected by the corpus callosum, that massive bundle of approximately 200 million nerve fibers we mentioned earlier, which allows constant communication between the two sides. This connection enables the hemispheres to work together seamlessly, coordinating their specialized abilities to produce unified, coherent thought and behavior. The story of hemispheric specialization begins in 1861, when French physician Paul Broca examined a patient who had lost the ability to speak, capable of uttering only a single syllable. After the patient's death, Broca discovered a lesion in the left frontal lobe and concluded that this region was essential for speech production. This area, now called Broca's area, provided the first clear evidence that the two hemispheres are not identical, that specific functions can be localized to one side of the brain. A decade later, German neurologist Karl Wernicke identified another left hemisphere region crucial for language comprehension. Patients with damage to Wernicke's area, located in the left temporal lobe, could speak fluently, but their speech made no sense, filled with meaningless words and phrases. They also couldn't understand spoken language. These discoveries established that for the vast majority of people, approximately 96% of right-handers and 70% of left-handers, language functions are lateralized to the left hemisphere. But the left hemisphere's specialization extends beyond language. It excels at sequential, analytical processing, at breaking complex tasks into components and processing them step by step. It handles mathematical calculations, logical reasoning, and the precise timing required for fine motor control. When you read these words, when you perform arithmetic, when you analyze an argument for logical consistency, you're relying heavily on your left hemisphere's specialized capabilities. The right hemisphere, by contrast, shows different strengths. It excels at spatial processing, at understanding relationships between objects in three-dimensional space, at recognizing faces and interpreting facial expressions. When you navigate through a room full of furniture, when you recognize a friend from across the street, when you appreciate a painting or visualize how furniture might fit in a room, your right hemisphere is taking the lead. The right hemisphere also plays a crucial role in processing emotional content, particularly negative emotions, and in understanding the emotional prosody of speech, the tone and inflection that convey feeling beyond literal meaning. Perhaps the most dramatic evidence for hemispheric specialization comes from split brain patients, individuals who had their corpus callosum surgically severed to control severe epilepsy. In the 1960s, neuroscientists Roger Sperry and Michael Gazzaniga conducted ingenious experiments with these patients that revealed how the disconnected hemispheres function independently. Because visual information from the left visual field goes to the right hemisphere and information from the right visual field goes to the left hemisphere. Researchers could present information to one hemisphere while keeping the other naive. When an object was shown in the left visual field, processed by the right hemisphere, the patient could not name it, because the language-dominant left hemisphere had no access to that information. Yet the patient could point to the object with their left hand, which is controlled by the right hemisphere. When asked why they pointed to that particular object, the left hemisphere, unaware of what the right hemisphere had seen, would confabulate, making up plausible-sounding explanations for behavior it didn't actually control. These experiments revealed that each hemisphere possesses its own perceptions, its own knowledge, its own decision-making capabilities. Yet we must be cautious about oversimplifying hemispheric differences. The popular notion of being left-brained or right-brained, of people being either logical and analytical or creative and intuitive based on hemispheric dominance, is largely a myth. In reality, virtually all cognitive tasks engage both hemispheres, which work together in intricate coordination. The hemispheres don't operate in isolation, but rather complement each other, with the corpus callosum ensuring that information flows between them, allowing integrated whole-brain processing. Recent research has revealed something even more nuanced. The two hemispheres don't just specialize in different functions, they specialize in different patterns of connectivity. The left hemisphere shows a bias toward intrahemispheric connections, forming tight networks within its own territory. The right hemisphere, by contrast, shows more interhemispheric connectivity, forming networks that span both hemispheres. This organizational difference may underlie the different cognitive styles associated with each hemisphere, with the left's local connectivity supporting focused, analytical processing, and the right's bilateral connectivity supporting holistic, integrative processing. The chemical messengers. Behind every thought, every emotion, every movement lies a complex ballet of chemical messengers called neurotransmitters. These molecules, released across synaptic clefts, are the fundamental medium through which neurons communicate. Understanding neurotransmitters provides crucial insight into how the brain generates our mental life and how that life can be disrupted in psychiatric and neurological disorders. Glutamate is the brain's primary excitatory neurotransmitter, used by more than 80% of neurons. When glutamate binds to receptors on a receiving neuron, it increases the likelihood that the neuron will fire an action potential. Glutamate is crucial for learning and memory, with the strengthening of glutamate synapses forming the basis of long-term potentiation. But too much glutamate can be dangerous. Excessive glutamate activation can cause neurons to fire so intensively that they die, a process called excitotoxicity that contributes to damage during strokes and some neurodegenerative diseases. Balancing glutamate is GABA, gamma-aminobutyric acid, the primary inhibitory neurotransmitter. GABA reduces neuronal excitability, preventing excessive firing and maintaining the delicate balance between excitation and inhibition necessary for normal brain function. Many anti-anxiety medications work by enhancing GABA signaling, increasing inhibition, and thereby reducing neural activity in anxiety-related circuits. The interplay between glutamate and GABA is fundamental to brain function, like an accelerator and brake working together to control the brain's activity levels. Dopamine, while less abundant than glutamate or GABA, plays outsized roles in motivation, reward, movement, and attention. The brain contains several distinct dopamine pathways, each serving different functions. The mesolimbic pathway, projecting from the midbrain to the nucleus accumbens, forms a crucial part of the brain's reward circuit. When you experience something pleasurable, eating delicious food, having sex, receiving praise, accomplishing a goal, dopamine neurons in this pathway fire intensely, creating feelings of pleasure and reinforcing the behaviors that led to the reward. This reward circuitry is hijacked by addictive drugs, which cause enormous surges of dopamine far exceeding natural rewards. Over time, the brain adapts to these artificial surges, requiring more of the drug to achieve the same effect, while finding natural rewards increasingly unsatisfying. Understanding dopamine's role in addiction has transformed how we view substance abuse, not as a moral failing, but as a medical condition involving disrupted brain chemistry. Dopamine also plays crucial roles in motor control through the nigrostriatal pathway, which connects the substantia nigra to the striatum. The death of dopamine neurons in this pathway produces the motor symptoms of Parkinson's disease. L-DOPA, a precursor to dopamine that can cross the blood-brain barrier, provides symptomatic relief by temporarily restoring dopamine levels, though it doesn't stop the underlying neurodegeneration. Serotonin modulates mood, anxiety, sleep, appetite, and numerous other functions. Most serotonin neurons are located in the raphe nuclei of the brainstem, but their axons project widely throughout the brain and spinal cord, allowing serotonin to influence virtually every brain region. Low serotonin function has been implicated in depression, anxiety, and obsessive-compulsive disorder, though the relationship is more complex than simple deficiency. Selective serotonin reuptake inhibitors, SSRIs like fluoxetine and sertraline, increase serotonin availability in synapses and provide relief for many people with depression and anxiety, though exactly how they work remains incompletely understood. Norepinephrine, closely related to adrenaline, plays key roles in arousal, attention, and the stress response. When you're startled or face a threat, norepinephrine floods your brain, sharpening attention, increasing vigilance, and preparing you for action. This arousal system is crucial for survival, but can become dysregulated in anxiety disorders and post-traumatic stress disorder, where the brain maintains heightened alertness even when threats have passed. Acetylcholine was the first neurotransmitter discovered and plays diverse roles throughout the nervous system. In the peripheral nervous system, it triggers muscle contraction at the neuromuscular junction. In the brain, it modulates attention, learning, and memory, with cholinergic neurons from the basal forebrain projecting widely to the cortex and hippocampus. The death of these cholinergic neurons contributes to the memory deficits in Alzheimer's disease, and medications that boost acetylcholine provide modest symptomatic benefits. Beyond these classical neurotransmitters exist numerous others, including endocannabinoids, which bind to the same receptors as THC from cannabis, endorphins, the brain's natural opioids that modulate pain and create feelings of euphoria, and various neuropeptides that fine-tune neural signaling. This chemical complexity allows for nuanced, flexible control of brain activity, enabling the rich variety of mental states we experience. The senses and the brain's construction of reality. Your experience of the world feels immediate and direct, as though you're perceiving reality as it truly is. But this is an elaborate illusion. What you experience as sight, sound, touch, taste, and smell are actually sophisticated constructions created by your brain from fragmentary sensory data. Understanding how the brain builds perceptual experience reveals both the power and the limitations of our access to reality. Consider vision, our dominant sense. Light enters your eyes and strikes the retina, where photoreceptor cells convert electromagnetic radiation into neural signals. But the retina doesn't simply transmit a raw image to the brain. It performs extensive pre-processing, detecting edges, movements, and contrasts before information even leaves the eye. These processed signals travel via the optic nerve to the lateral geniculate nucleus of the thalamus, which serves as a relay station, and then to the primary visual cortex in the occipital lobe. The primary visual cortex is organized in remarkable ways. Different neurons respond selectively to specific features of the visual scene. Some fire only when they detect vertical edges. Others respond to horizontal edges, still others to specific orientations in between. Some neurons detect motion, others respond to specific colors. This feature detection is just the first step. From primary visual cortex, information flows into higher visual areas that recognize increasingly complex patterns, from edges to shapes, to objects, to faces, to entire scenes. This hierarchical processing culminates in your conscious visual experience, but that experience is far from a simple representation of what's out there. Your brain fills in the blind spot where the optic nerve exits the retina, creating the illusion of continuous vision where no photoreceptors exist. It stabilizes vision despite constant eye movements, constructs three-dimensional depth from two-dimensional retinal images, and maintains color constancy so that objects appear the same color under different lighting conditions. What you see is not reality itself, but your brain's best hypothesis about reality, constructed from limited data and shaped by your expectations, attention, and prior knowledge. Hearing operates through similarly sophisticated processes. Sound waves vibrate your eardrum, which transmits these vibrations through tiny bones to the fluid-filled cochlea. Within the cochlea, different hair cells respond to different frequencies, creating a tonotopic map of sound frequencies. These signals travel through the auditory nerve to the cochlear nucleus in the brainstem, then through several relay stations before reaching the primary auditory cortex in the temporal lobe. The auditory cortex analyzes sounds for their pitch, timbre, location, and temporal patterns. Specialized regions process speech sounds, music, and environmental noises. The brain performs remarkable feats of auditory processing, separating one voice from the background noise at a cocktail party, recognizing a familiar voice on the phone, determining that a series of different sounds constitute words in a language you understand. All of this happens effortlessly, automatically, below the threshold of conscious awareness, until suddenly you experience hearing, the subjective quality of sound constructed by your brain. Touch, pain, temperature, the somatosensory modalities are processed through yet another pathway. Receptors in your skin detect pressure, vibration, temperature, and tissue damage, sending signals up the spinal cord to the thalamus and then to the somatosensory cortex in the parietal lobe. Like the motor cortex, the somatosensory cortex is somatotopically organized, creating a sensory map of your body with disproportionately large representations for sensitive areas like the hands and face. But even simple touch is far from simple. Your brain must integrate information about texture, temperature, pressure, and location to create the unified sensation of touching an object. It must distinguish between touches caused by your own movements versus touches from external sources. It must continuously update its representation of your body's position in space based on sensory feedback from muscles and joints. All of this integration happens automatically, giving you a coherent sense of your body and its interactions with the world. The emotional brain. Emotions are not peripheral to cognition, but central to it. Far from being opposed to reason, emotions guide attention, motivate behavior, facilitate learning, and enable social connection. Understanding the neural basis of emotion reveals how deeply feelings are woven into the fabric of mental life. The limbic system, a collection of interconnected structures deep within the brain, plays crucial roles in emotional processing. The amygdala, a small almond-shaped structure, serves as the brain's threat detection system. It responds rapidly to potential dangers, triggering fear responses before conscious awareness even registers the threat. When the amygdala detects danger, it activates the hypothalamus, which triggers the stress response. Increased heart rate, rapid breathing, release of stress hormones like cortisol and adrenaline, heightened attention, and vigilance. This rapid threat response evolved to keep our ancestors alive in a dangerous world. Better to jump at a shadow that might be a predator than to fail to react when danger is real. But in modern life, this sensitive threat detection system can become problematic. Anxiety disorders often involve an overactive amygdala that sees threats where none exist, triggering fear responses to situations that pose no real danger. Post-traumatic stress disorder involves a hypersensitive amygdala that has been conditioned by trauma to respond intensely to reminders of past danger. The hippocampus, sitting adjacent to the amygdala, is crucial for encoding memories, particularly emotional memories. Emotionally significant events are remembered far better than neutral ones because the amygdala signals the hippocampus that an experience is important and should be strongly encoded. This is why you likely remember exactly where you were during major life events, your wedding, the birth of a child, receiving life-changing news, while countless mundane days blur together and fade. The prefrontal cortex regulates emotions, providing top-down control that can modulate the amygdala's responses. When you consciously decide to stay calm in a stressful situation, when you reappraise a negative event to see it in a more positive light, when you suppress an emotional outburst, you're using prefrontal cortex to regulate limbic activity. Depression and anxiety disorders often involve impaired emotion regulation, with reduced prefrontal control allowing limbic structures to dominate, creating persistent negative emotional states. The insular cortex provides awareness of your body's internal states, your heartbeat, your breathing, the sensations in your gut. This interoceptive awareness forms the basis of emotional feelings. When you feel anxious, you're aware of your racing heart and shallow breathing. When you feel disgust, you're aware of nausea. When you feel excited, you notice increased energy and arousal. The insula integrates these bodily signals with cognitive appraisals to create the subjective feeling of emotion. Emotions also involve neurotransmitter systems we discussed earlier. Dopamine drives motivation and pleasure. Serotonin modulates mood and anxiety. Norepinephrine creates arousal and vigilance. Oxytocin and vasopressin, often called social hormones, facilitate bonding, trust, and attachment. The complex interplay of these chemical systems, combined with activity in emotion-related brain structures, generates the rich emotional life that colors every experience and guides every decision. The social brain. Humans are profoundly social creatures, and our brains reflect this. We possess specialized neural machinery for navigating the social world, for recognizing faces, interpreting emotions, understanding others' intentions, cooperating with allies, and competing with rivals. These social cognitive abilities may have been the primary driver of human brain evolution, with intelligence evolving not primarily to solve ecological problems, but to navigate complex social relationships. The fusiform face area, a region in the temporal cortex, responds selectively and powerfully to faces. We can recognize thousands of individual faces, distinguish subtle facial expressions, determine where someone is looking, and judge age, sex, and emotional state from facial information alone. This remarkable face processing ability emerges early in development. Newborn infants preferentially look at face-like patterns and becomes highly specialized through experience. Damage to the fusiform face area produces prosopagnosia, or face blindness, where people lose the ability to recognize faces while retaining normal object recognition. Some prosopagnosics can't even recognize their own face in a mirror, though they can identify people by voice, gait, or context. This selective deficit demonstrates that face processing relies on specialized neural machinery distinct from general object recognition. Theory of mind, the ability to understand that others have minds, with their own beliefs, desires, and intentions, represents another crucial social cognitive capacity. When you predict how someone will react to news, when you recognize that someone holds a false belief, when you attribute mental states to explain behavior, you're using theory of mind. This ability emerges in childhood, typically around age 4, when children begin to understand that others can hold beliefs different from their own or different from reality. Theory of mind involves a network of brain regions, including the temporoparietal junction, the medial prefrontal cortex, and the posterior superior temporal sulcus. Autism spectrum disorders often involve impairments in theory of mind, making it difficult to intuitively understand others' mental states and leading to challenges in social interaction and communication. Yet many autistic individuals develop explicit, systematic approaches to understanding social behavior, demonstrating that multiple routes to social cognition exist. Mirror neurons, discovered in the 1990s, fire both when an animal performs an action and when it observes another animal performing that same action. These neurons may provide a neural basis for imitation, empathy, and understanding others' actions. When you watch someone reach for a cup, mirror neurons in your motor cortex activate as though you were performing that action yourself. This neural mirroring might allow you to understand others' actions by simulating them internally, providing an embodied route to social understanding. Empathy, the ability to feel what others feel, engages many of the same neural circuits involved in experiencing emotions firsthand. When you see someone in pain, your own pain-related brain regions activate. When you observe someone receiving a reward, your reward circuits engage. This shared neural representation of others' experiences may be the biological foundation of empathy, allowing us to literally feel with others and motivating pro-social behavior. The gift of mystery. As you settle more deeply into relaxation, as sleep approaches and consciousness begins to dissolve into dreams, consider the magnificent paradox we've explored tonight. The brain that allows you to contemplate the universe is itself a part of that universe, a product of the same physical laws that govern stars and atoms. 3 pounds of cells assembled through billions of years of evolution, refined through your individual development, shaped by every experience you've ever had, generating this moment of awareness, this sensation of being you. The brain is not merely a tool you use to navigate the world. It is the source of the world you experience, constructing reality from fragmentary sensory inputs, creating the coherent narrative of your life from countless neural processes you never directly perceive. The colors you see exist nowhere but in your brain's interpretation of electromagnetic waves. The sounds you hear are your brain's rendering of pressure waves in air. The self you take to be the central character in your life story is itself a construction, an emergent pattern in neural activity that creates the illusion of a unified, continuous agent persisting through time. Yet calling the self an illusion doesn't diminish its reality in the only way that matters, your subjective experience. You are real. Your thoughts are real. Your feelings are real. They are what your brain does, how it feels from the inside to be this particular configuration of neurons firing in these particular patterns at this particular moment in space and time. The mystery of consciousness, the hard problem we explored earlier, may never be fully solved. Perhaps we will always face an explanatory gap between objective descriptions of neural processes and subjective experience itself. Perhaps consciousness requires concepts we haven't yet developed or represents a fundamental feature of reality not reducible to simpler components. Or perhaps future neuroscience will crack this problem as molecular biology cracked the mystery of life, and consciousness will be demystified without being diminished. Whatever the future holds, we can be certain that our understanding of the brain will continue to deepen. New imaging techniques will let us observe neural processes at unprecedented scales and resolutions. Advanced artificial intelligence may help us decode the brain's computational principles. Stem cell research and gene therapy may offer new treatments for currently incurable brain diseases. Brain-computer interfaces may extend cognitive capabilities beyond biological limits, blurring the line between mind and machine. These advances will raise profound questions. If we can upload memories to computers, what becomes of the self? If we can enhance intelligence through neural implants, should we? If we can manipulate the neural basis of emotions and desires, does free will become meaningless? These aren't abstract philosophical puzzles. They're practical ethical challenges that neuroscience will force us to confront in the coming decades. As you drift now toward sleep, your brain will continue its endless work. It will consolidate the memories formed today, replaying neural patterns to strengthen synaptic connections. It will cycle through stages of sleep, each serving purposes we only partly understand. It will generate dreams, those strange nocturnal narratives that mix memory fragments with impossible scenarios. And it will prepare for tomorrow, maintaining readiness to wake, to perceive, to think, to be you once again. The human brain, most mysterious object in the cosmos, most intimate aspect of your existence, the source of everything you think and feel and know. 3 pounds of tissue generating an infinity of experiences, forever exploring the universe while simultaneously exploring itself, asking questions about its own nature that it may never fully answer. It is ancient in its evolutionary heritage, yet unique in each individual. It is exquisitely organized, yet constantly changing. It is biological, yet it produces thoughts that seem to transcend biology. It is matter, yet it gives rise to mind. Tonight, we have journeyed through the landscapes of neuroscience, exploring neurons and synapses, energy metabolism and cortical organization, consciousness and neuroplasticity, evolution and disease. We have confronted the limits of current understanding and stood in awe before questions that remain unanswered. We have seen that the brain is simultaneously the tool we use to understand the world and the greatest mystery that world contains. The brain you possess right now, generating these thoughts, processing these words, creating this moment of conscious experience, is the result of an unbroken chain of evolution, stretching back billions of years, refined through your individual development, shaped by every experience you've accumulated, poised to continue changing with every moment you live. It is capable of appreciating beauty, of solving problems, of feeling joy and sorrow, of imagining possible futures and learning from past mistakes. It can ponder the origins of the universe and the nature of consciousness itself. It can create art and music, can build civilizations and explore other worlds, can love and hope and dream, and remarkably, wonderfully, mysteriously, it is you. Not merely the thing that contains you or produces you, but in the deepest sense, it is you. The brain reading these words is not separate from the mind experiencing them. They are two ways of describing the same phenomenon, one from the outside, one from the inside. You are your brain, and your brain is you, experiencing itself, exploring itself, forever seeking to understand the mystery at the heart of its own existence. If you found this exploration of the human brain thought-provoking and restful, please consider subscribing to DREAMLAB Knowledge. Hit that like button to help others discover this journey through the most complex and mysterious object in the cosmos. Share this with anyone fascinated by neuroscience, consciousness, and the profound questions at the intersection of mind and brain. Your engagement helps us continue producing content that delves deep into scientific mysteries while creating a peaceful space for reflection and wonder. Together, we can build a community that values both knowledge and contemplation, that embraces complexity while finding calm, that sees science not as a collection of dry facts, but as a profound meditation on the nature of reality and our place within it. Thank you for joining this journey into the magnificent mystery of the human brain, and until next time, keep questioning, keep wondering, and keep marveling at the extraordinary 3-pound universe residing within your skull.